These are your notes on meiosis. Remember, mitosis is the process of making body cells. Meiosis is the process of making sex cells, so either sperm cells or egg cells. Remember, mitosis was PMAT, prophase, metaphase, anaphase, telophase, just one time. Meiosis is PMAT 2 times, so you have 2 rounds of cell division. It's division of the nucleus again, just like mitosis, except the purpose here, at least in mammals and humans, is to make reproductive cells, sperm cells and egg cells. It makes 4 haploid cells. That means I think of haploid as half. It just has one set of chromosomes. Your cells, your body cells, all have 2 sets of chromosomes. You get 23 chromosomes from your mom, like this picture here, from your mom, your egg cell, and you get 23 chromosomes from your dad in the sperm cell. And so that makes a zygote with 46 chromosomes. All of your body cells have 46 chromosomes, a set from mom and a set from dad. But these sperm cells and egg cells, they only have 23 chromosomes. So that every time we make a person, they only have 46 chromosomes. We don't double the number of chromosomes every single generation. People can't do that. Some plants have multiple sets of chromosomes, but not people. This makes, the other purpose of meiosis is to make unique genetic combinations. Remember that mitosis makes an identical cell, hopefully. It's exactly like the parent cell. But meiosis creates variation. There are 2 main ways that this happens, through independent assortment and crossing over, which we'll talk about. And then down here at the bottom, it goes through PMAT 2 times. So it's prophase, metaphase, anaphase, telophase 1, and then prophase, metaphase, anaphase, telophase 2. Some people get fertilization confused with meiosis. So meiosis is the process of making the sperm cell and the egg cell. Fertilization is when the egg cell and the sperm cell come together and make the zygote, or the fertilized egg. So remember, the purpose with meiosis is that we get half the number of chromosomes here in the egg cell, half the number in the sperm cell, so that the zygote has 46 chromosomes. Here's a little overview of meiosis. The first thing that has to happen, just like in mitosis, is DNA replication. The really simple cell here, we're starting with 2 chromosomes. They do DNA replication, and the chromosomes are copied. So now we've got sister chromatids. Remember that word? In the first round of division, you split these homologous chromosomes. Okay, so remember, these are identical copies. These are sister chromatids. But let's say this chromosome is from mom, this one, and this chromosome is from dad. Okay, but they have the same or similar types of information. Like these have genes for eye color, and this has genes for eye color. Maybe you got green eyes from your mom, and you got brown eyes from your dad. In the first round of division, these homologous chromosomes separate. So now you can see we have already reduced the chromosome number by half. Here we had 2 colors, 2 chromosomes, and here we just have 1 color in each cell. So we already have half the number of chromosomes. Then the second round of division is a lot like mitosis. We split the sister chromatids into different cells. So here we already have half the number of chromosomes. The second round of division will split the copies. Okay, so let's go through all of that in steps. Interphase 1, just like before mitosis, the DNA is going to replicate. That's during S phase. Remember, you have G1, normal cell processes take place. You have S, where you have DNA replication, and then G2, where the number of organelles pretty much doubles. All right, and then you can go into meiosis 1. The goal here is to separate homologous chromosomes and reduce the chromosome number by half. Here are the steps. So prophase 1, this is where homologous chromosomes have to find each other. This is totally different than mitosis. Mitosis Remember, the nuclear membrane goes away, the chromosome starts to coil up into the form of chromosomes, the spindle fiber forms. All of that stuff still is happening here. So I'm expecting you to know that that still happens. But the difference is in meiosis, you have these homologous chromosomes that have to find each other. So you have chromosome from dad has to find chromosome from mom. You have 23 pairs of chromosomes. That means chromosome number 1 from dad has to find chromosome number 1 from mom. Chromosome number 2 from dad has to find chromosome number 2 from mom. That's a lot of matching up. So the new word for that is synapse, when those come together. The other thing that can happen when those chromosomes come together is that they can cross over. So they can actually switch little pieces of their chromosome. So you can see that the colors switched places. This is not a translocation when we talked about mutations. So here, there's nothing bad that has happened, because you still have all the same genes, all the right genes. You've just switched up, like maybe which type of gene is where. So you're not losing any information, and you're not getting information in the wrong place. So here's another picture of that. So that we're non-sister chromatids. So they're not identical, but they do have the same genes. So their genes are letters here. So we've got gene J, K, and L on mom's chromosome. Maybe this pink one is mom, and this JKL on dad. They overlap and they swap spots. So then the end version here is that you have the lowercase, the recessive genes here, and you've got the dominant genes here. So just so happens in my picture here that dad has all the dominant copies of genes and mom has all the recessive copies of genes. It doesn't really work like that, but it makes the picture easier to understand. So then dad's chromosome is now over here with all the dominant genes on it, and mom's chromosome is now over here with all these recessive genes on it. But you've got a brand new chromosome that never existed before. That's part of what makes us all unique, this shuffling around of the chromosomes crossing over. And this is all just in prophase 1. All the homologous chromosomes have to find each other in synapse, and then they have to cross over. And at the same time, all the other steps that we talked about in mitosis are still happening, like the spindle fibers forming, the nuclear membrane goes away, the chromosomes condense and find each other. Metaphase 1, you're still thinking metaphase middle, but here you have to remember that it's the homologous pairs of chromosomes that are lined up at the equator. We don't have a single line of X's. We've got a double line of X's. Metaphase 1 is where independent assortment can happen. You do not have all of mom's chromosomes lined up on one side and all of dad's on the other. You could. That's pretty unlikely when you have 23 pairs of chromosomes, but in this picture, it's a little bit more likely. Here on this side, I've got mom's on this side and dad's on this side, but it doesn't have to be that way. So here, the bottom is mom's on the left and dad's on the right, but up here we've got dad's on the left and mom's on the right. So that combined with the crossing over is going to create daughter cells that have a unique combination of chromosomes. Here's another picture of independent assortment with the same 2 cells to start with. Dad's chromosomes here, mom's here, then dad's here, and then here I've got mom's on this side and dad's on this side. So you can see if both these go through meiosis, they split once and they split again, you end up with 4 sperm or 4 egg cells. And you've got different combinations. So you've got blue and pink. This is also blue and pink. And then you've got a big pink and a little blue and a big pink and a little blue. But if the chromosomes line up differently, now you've got your pink chromosomes together on those 2 cells, and then you've got your blue in these 2 cells. The more chromosomes you have, the more opportunity that gives you for different combinations during independent assortment. If in my picture here, I've got 6 different chromosomes, 6 different chromosomes. You can figure out how many ways you can arrange them if you do this little equation here. If you take 2 to the nth power, where n is the haploid number. So here I have 6 chromosomes. That's my diploid number. So half of that would be 3. So if I take 2 to the 3rd power, that gives me 8 different combinations. So here they're showing you those 8 different ways you can line up the red and the blue. So if you look at those closely, they should all be different. But this gets big really fast. In people, you have 2 times or 2 to the 23rd power. That gives you over 8 million different ways that the chromosomes can line up. Lots of genetic diversity. Okay, so let me go back a little bit. Prophase 1, homologous chromosomes came together. You had crossing over. Metaphase 1, the homologous chromosomes lined up at the equator, randomly, independent assortment. And now we've got anaphase 1. Homologous chromosomes are going to separate. They're going to move toward the poles. Telophase 1 and cytokinesis. So now we've got a haploid number. We've got half the set of chromosomes. And then we're going to have cytokinesis. We're going to start to split into 2 daughter cells. I'm skipping some of the details. They're the same as mitosis. So like the nuclear membrane starts to come back, the spindle fibers are going to go away. But now, instead of that being it, we're going to keep going. We've got meiosis 2. The goal of the second half is to separate the sister chromatids, separate the copies. This half is a lot like mitosis. There's no interphase 2, or if there is, it's really short. There's no DNA replication because we already have copies. We don't need another copy. Prophase 2, it's the same as prophase in mitosis. It's just happening in 2 cells at the same time. Spindle fibers show up, nuclear membrane goes away. The chromosomes are already pretty condensed because we didn't go back into interphase. Metaphase 2 looks like mitosis, got a single line of chromosomes. Anaphase 2, the sister chromatids are going to separate. Telophase 2 and cytokinesis. Now we're going to make those 4 haploid daughter cells. The fancy word for that is gametes, or sex cells, sperm cells, and egg cells. Spermatogenesis is the word for making sperm cells. So you start with the 46 chromosomes. Remember, the first round division cuts it down to the haploid number, and then you separate sister chromatids, and you can make 4 sperm cells. Oogenesis is the process of making egg cells. You don't have to know this, but let's see. So you start with 46 chromosomes. You divide that in half and get 23. But then in the second round of division, the cytoplasm is not split evenly. So more cytoplasm is going to go to one cell than the other. And then more cytoplasm is going to go to this cell than the other cells in the second round of division. So with oogenesis, you actually only make 1 egg. And these other 3 are going to be polar bodies.